A very wonderful afternoon, respected faculty and friends. I am Priyall Narwal, and today I'll be presenting my project on molecular docking of the FUS protein, which is associated with ALS. ALS stands for amyotrophic lateral sclerosis, that is a fatal and a progressive neurodegenerative disease that affects motor neurons. The mutations in FUS protein causes abnormal aggregations inside neurons, which leads to neuron death. Therefore, targeting FUS protein with small molecules can be a promising approach for ALS treatment. FUS protein background. FUS stands for fused-in sarcoma. It is an RNA-binding protein mainly present in the nucleus of cells. FUS is important for making RNA from DNA, RNA splicing, DNA repair, and transporting RNA inside the cells. FUS proteins has different domains: RNA binding domain, nuclear localization signal (NLS) that helps it enter the nucleus, low complexity region that helps protein interactions. Mutations in FUS are linked to ALS, frontotemporal dementia, and some cancers. When mutated, FUS accumulates in the cytoplasm and forms toxic aggregates. The aim of my project was to identify potential and small molecule inhibitors for FUS protein using molecular docking. Objectives were to download FUS protein structure, to select ligands from literature, to perform docking using PyRx, and to analyze interactions using Discovery Studio and PyMod. 5 selected ligands based on literature and neuroprotective properties: ATP, nimbin, dihydroxymethylphasin, curcumin, chloroindocin C. These compounds were chosen because they were reported in research papers to interact with neurodegenerative pathways or protein aggregations, and the IDs are mentioned beside them. Then protein structure was actually downloaded from RCSB PDB with ID 6PXV. Ligands were downloaded from PubChem in 3D SDF format, and the softwares used are PyRx, AutoDock, and Discovery Studio. For methodology, firstly, the FUS protein structure was downloaded and cleaned by removing water and unwanted molecules. Then ligands were downloaded and energy minimized using OpenBabel and PyRx only. All molecules were converted to PDB-QD format for docking. Docking was performed using AutoDock Vina and PyRx. A grid box was actually created around the active sites of FUS protein. Binding affinity values were recorded for all the ligands. This is the grid box formation, and this is the running of Vina wizard. And the results were analyzed. Nimbin showed the best binding affinity, that is minus 5.6 kilocalories per mole. Then dihydroxymethylphasin, that is minus 4.5, curcumin minus 4.5, and ATP and chloroindocin C showed lower affinity. The affinity value ranges between moderately acceptable values. But we'll be using this only because although the docking score is actually very moderate, it is important to note that FUS is an intrinsically disordered RNA binding protein, which often lacks rigid binding pockets, leading to comparatively lower docking scores than enzyme targets. Therefore, the observed binding affinity is considered acceptable for preliminary screening studies. The natural origin and reported neuroprotective properties of nimbin further support its selection as a potential lead document. So the best candidate here goes to nimbin. Then methodology for docking. The docking was performed using AutoDock Vina and PyRx. No. Okay, only then the best selected candidate was actually taken. The best docking pose was selected using the Vina split option. Then the protein ligand complex was visualized in Discovery Studio. This is the docking done in BioVia. The yellow highlighted portion is our protein, and the other one is our ligand. The ligand interaction can be seen. There's a green dash lines that basically goes for our interactions between our ligand and protein. This is the 2D interaction. These interactions confirm a stable ligand binding. More negative binding energy indicates stronger interactions, and the natural compounds like nimbin showed a neuroprotective potential. Docking only predicts interaction and does not confirm the real biological activity. There are certain limitations to it, that is, it is only computational. FUS is actually more flexible and partially disordered. We only tested 5 ligands, and toxicity and blood-brain barrier penetrations were not studied. Then moving on to the conclusion, molecular docking was performed on the FUS protein, which is a key target in ALS among 5 ligands. Nimbin showed the best binding affinity and the stable interaction, and we told you why this was the best candidate, and this suggests that nimbin may act as a potential inhibitor of the FUS protein, and however the further experimental validations is required to confirm its therapeutic potential. Thank you for your attention.